The following program is part of a college course. These telecourses allow you to earn college credits at home. For more information, contact a college or university near you.
Between atomic energy, damaging DNA, and creating monsters, such as giant ants or giant rabbits, in some ways is very similar to the concerns today about genetic engineering and creating monsters. Look, for example, at the book and the movie Jurassic Park. This was recombinant DNA technology gone awry. And while most of it was really quite far-fetched, the basic principles were in fact quite reasonable.
In each of our cells lies the secret of our heredity, DNA. In these intricate molecules, nature stores the genetic instructions that build all living things. Today, we have the knowledge and technology to alter these instructions. But what could happen if we tamper with this molecule of life? And if lives are at stake, can we afford not to? In the mid-19th century, war ravaged Europe. By 1869, one of the world's bloodiest decades was drawing to a close. Ironically, the misery born in the battles of that time would lead to one of biology's most important discoveries. Supplied with bandages from wounded soldiers, a Swiss chemist named Friedrich Miescher isolated a molecule from the nuclei of white blood cells. Miescher named the chemical nuclein, which became known as deoxyribonucleic acid, DNA. Unaware that Miescher had just discovered the blueprint of life, scientists largely ignored the work. The structure and function of this molecule would remain a mystery for another 80 years. (1952), big cars and modern appliances. With World War II behind them, scientists applied their wartime technology to peaceful uses, including their quest to find the genetic material of life. Although challenged by some, many scientists believed that proteins, not DNA, held the secrets of heredity. Then two microbiologists, Alfred Hershey and Martha Chase, made an extraordinary discovery. Using radioactive isotopes, they traced the DNA of certain bacterial viruses. Ironically, using an ordinary household blender to separate the viruses from their bacterial hosts.
We tried various things that didn't work until we got this elegant little machine, which was invented, I think, to make cocktails. And it worked right away. It worked fine.
In this pivotal experiment, Hershey and Chase proved that the genetic material of the virus was not a protein, it was DNA. Now the quest to determine the structure of these molecules reached a feverish pitch. In 1953, James Watson, a young colleague of Hershey's, teamed up with a former physicist named Francis Crick. Inspired by X-ray crystallography, which revealed a spiral pattern, they pieced together a stunning three-dimensional model for DNA, the double-stranded helix.
I think the discovery of the structure of DNA is really the fundamental basis on which is all modern molecular biology is established. the significance of which is almost unfathomable. So it was perhaps, to my mind, probably as seminal as the discovery by perhaps Darwin, the laws of survival, or Mendel's law of inheritance.
In recognition of their landmark contribution, Watson and Crick received the Nobel Prize for Medicine and Physiology in 1962.
Well, the outcome of simplicity was the complementary relation of the bases, that the sequence of one chain, the sort of alphabet on one chain determined the alphabet on the other. So if you had A on one chain, adenine, you had to have thymine on the other chain. So and just a pretty molecule.
The basic components of DNA are the 4 different letters of the alphabet, which we call nucleotide bases, represented by A, G, C, and T. And the remarkable property about these bases is they pair together, the A's always matching up with the T's and the G's matching with the C's, to create then a double-stranded DNA molecule where the matching bases are the individual stairs as one climbs up the spiral of life.
DNA contains 2 strands of building blocks called nucleotides, arranged like a spiral staircase. Each nucleotide includes 3 parts: a phosphate group, a sugar molecule, and one of 4 bases: adenine, guanine, cytosine, or thymine. The sugar phosphate bonds form the double backbone of the molecule, the handrails of the staircase. But we find the genetic key to DNA in the steps of this stairway, the nitrogen-containing bases. These bases link up using hydrogen bonds in a very specific way. Adenine will bond only with thymine, A to T. Cytosine only bonds with guanine, C to G. While these basic pairings never change, the order of the pairs along each strand varies greatly from one species to the next. In this elegant design, we see how nature stores the instructions to build all living things.
In science, good models offer predictive value. Using their new design, Watson and Crick could now suggest an answer to one of biology's long-standing mysteries: how our genes replicate before the cell divides.
When one thinks about DNA replication, one has to think about a machinery which must copy itself exactly for the entire adult life of an individual. If you think about the number of cells that we make in the immune system, for example, we make billions of cells every day. And the idea that there's a machine which can duplicate itself hour by hour, minute by minute, and to be precisely copied, because if it is not, it has the potential to cause havoc on the cell.
To make perfect copies of our DNA, an enzyme first unwinds the 2 strands of the molecule by separating its base pairs. Another enzyme then attaches free nucleotides to the exposed sites. The existing strand determines the composition of the new strand. According to the base pairing rules, adenine bonds with thymine and guanine with cytosine. In long strands of DNA, replication occurs in many regions simultaneously, forming the sister chromatids required for cell division.
The structure of DNA revealed the secrets of its function and launched a new field of biology.
Without the knowledge that the DNA had a structure which was helical, there would have not been possible almost any other experimentation in terms of how proteins are generated, how DNA replication occurs, how genes are transferred downstream, how genes can be manipulated, how DNA can be manipulated, how DNA can be sequenced.
Today, with their growing knowledge, scientists can alter the genetic makeup of living things. While this may frighten some, for others it offers the only chance for a normal life. Doctors knew right away that young Andrew Gobia had severe combined immune deficiency, the genetic illness known as SCID. Even before Andrew was born, doctors found the boy would be healthy except for one crucial factor. He was missing a tiny fragment of DNA that directed the production of white blood cells. The result would be an immune system so weak that even a childhood flu could endanger Andrew's life, as it had taken his sister before him.
Andrew's sister also had the same immune deficiency that he had. She inherited the same 2 defective genes. The problem is the first child in a family with this disease often doesn't come to medical attention until they have a severe infection. She didn't come to our hospital for a bone marrow transplant until she already had a pneumonia that proved to be fatal. So Andrew benefited from that because we knew, because of his sister, to check in his pregnancy to see if he had this disease.
Until recently, some children with immune deficiencies had to undergo expensive and painful bone marrow transplants. Others were forced to live in sterile plastic enclosures to prevent infection. Today, using our understanding of DNA, scientists are attempting to replace the defective fragments that cause some genetic ailments in a process known as gene therapy.
The physician has to identify the defect for which gene therapy can be considered. And at that point, the physician will have to come in contact with the basic scientist who is actually involved in this kind of research.
By DNA analysis, we were able to determine that Andrew had, in fact, inherited 2 broken copies of the ADA gene. So that predicted that he would have this immune deficiency problem when he was born.
Doctors decided to attempt a replacement of the defective segment of Andrew's DNA, the ADA gene. But to do this, they would need copies of a normal gene.
To be able to perform gene therapy for Andrew, we needed to have the gene that he was missing, have a normal copy of that, and have a way of getting it into his body. And the normal gene that he was missing, ADA, had been cloned many years ago and was available in a virus or vector that could carry it into his cells.
In gene therapy, cloning is the process of reproducing copies of a normal gene or DNA strand. Doctors then deliver these copies to the patient using vectors.
Vectors are basically delivery vehicles, and the real challenge and the barrier in gene therapy is once we've cloned a normal gene, how do you get it into cells? And the way you get it into cells is you put it into a vector. In most cases, the vector are viruses that normally infect cells that we can engineer to carry a gene of therapeutic interest into the cell.
In Andrew's case, the missing DNA segment was attached to a virus that carried the DNA into his blood and to his bone marrow.
The process of making a vector starts with these viruses being cloned. Just like you clone a normal gene, you can clone the genes for a virus. And once you've cloned it, you now have a DNA form that you can modify in the laboratory using restriction enzymes. So you can cut open the virus, clone in a human gene, and put it back together.
To clone a gene, scientists often isolate it from a healthy cell's chromosomes using special proteins called restriction enzymes. Each different enzyme recognizes a different site on the DNA. Like a pair of molecular scissors, the enzyme cuts the DNA, exposing a specific sequence of bases. Applied to the DNA of a vector, such as a virus, the same enzyme reveals the same sequence of base pairs, what scientists call sticky ends. When the two DNA sources are mixed, the sticky ends pair up, A to T and C to G. A joining enzyme called a ligase then reseals the double helix. We call the result recombinant DNA, a molecule ready for cloning or transport to a new host cell.
Once doctors had isolated the new gene and delivered it to Andrew, they faced yet another challenge: how to measure their success.
Is that the new blood sample from Andrew?
Yes, it is. Mm-hmm.
Oh, good. So we'll have some results to look at in the morning?
You betcha. Okay.
Bye-bye.
All right. Have a nice evening.
The way we're measuring how successful our gene transfer has been to get blood samples from Andrew sent to us once a month and look in the DNA of his white blood cells and ask, is our gene there? And the way we're looking is by doing PCR. And using PCR, or polymerase chain reaction, we can amplify the rare cells in there, that the DNA in rare cells that might have our gene, and actually, in a quantitative manner, find out how many cells in his blood have the unique sequences that we put in as the normal ADA gene.
To Andrew Gobia and the many thousands of people who suffer from genetic illness, gene therapy offers the promise of a normal life. But this therapy cannot yet treat all genetic diseases.
When somebody goes to the hospital for any kind of a disease, usually there's a culmination, even for cancer, a combination of chemotherapy, radiation therapy, tumor removal. It will still be, gene therapy will still be used for a while as an adjunct to what is going on, or a replacement for something, but will not be complete replacement until we are absolutely certain that this form of medication is working. So the important things that the physician and the scientist have to come to understanding is that, is this the best course for the patient?
We're hoping that it will mean that he will be able to make genetically cured cells for the rest of his life. We're hoping that we have basically eliminated his immune deficiency by giving him back the gene that he was missing.
2 years have passed since doctors began this life-saving experiment, and they say that little Andrew's chances of living a long and healthy life are very, very good. To prevent the suffering of genetic illnesses, scientists need to increase the availability of normal human genes. But first, they have to find them.
To perform gene therapy, the first step is to have the gene that you need to put into a patient. And that's the process of gene cloning. The way in which you isolate a human gene is either to know something about the gene you're looking for and find it from among all the genes in a patient's cells, or to know where a broken gene is on the chromosome and then look in that neighborhood.
Searching for a gene presents a daunting challenge. Scientists must scan through billions of base pairs, essentially the entire chemical memory of our DNA, in order to isolate a specific gene. Imagine searching for a single page in a single book in a library that has no index.
The 23 pairs of chromosomes that are present in each and every human cell are essentially the blueprint for life. So they have all the information that's necessary for starting you out as a single cell and carrying you through this marvelous process of development where you end up being an organism with 10 to the 14th cells. That's a 1 followed by 14 zeros, an astronomical number.
One chromosome represents an entire huge long single double-stranded DNA molecule from start to finish. In fact, if you were to take all of the DNA in human chromosomes and stretch it out and put it end to end, it would measure about 6 feet, which is extraordinary since every cell in the human body contains this complete DNA in its nucleus.
Chromosomes also contain useful proteins, which act like microscopic spools, helping to wind up the many thousands of base pair sequences that make up our DNA.
It's basically everything we can learn about the basic component that makes humans humans, the DNA content. And it's the equivalent to having every single letter in an entire library from start to finish, an entire collection of books in the library.
The quest to sift through our genetic library, perhaps 100,000 genes in all, is called the Human Genome Project.
Looks like they're about 6 megabases apart.
Gee, it's a lot closer than we thought they were.
Scientists all over the world are cooperating in this molecular gene hunt. One of these scientists is Dr. John Wasmuth.
Sounds good. Should have a map by Thursday. See you later. And we focus on chromosome number 5, which represents about 5% of all the DNA in the human genome. There are, in fact, ways to physically separate one chromosome from another.
Scientists sometimes use lasers to analyze and isolate chromosomes. However, Dr. Wasmuth's group uses chemical tagging to map individual DNA fragments.
Mapping the location of any piece of DNA is based on the fact that the DNA double helix is complementary. If you look at the way DNA base pairs, G with C and A with T, and you have a stretch of, let's say, 30,000 base pairs, and you unwind it and separate the two strands, you then have a cloned piece of DNA which is complementary to either one of those two strands. It will basically seek out its exact partner, and using various tags on the cloned DNA, whether it be radioisotopes or fluorescent tags, you can identify the exact place where that cloned complementary piece of DNA finds its brother or sister. Take a picture of the gel? Yep. See how it looks.
John Wasmuth's team of molecular detectives are hot on the trail of many genetic ailments. They have already found a marker for the gene that causes Huntington's disease, a fatal degenerative illness that attacks the brain. While their work has just begun, it promises the ultimate weapon against the threat of inherited disease.
I think the Human Genome Project and gene therapy are very closely interrelated. We need to have the genes responsible for disease to be able to treat a patient. And so until we know what gene is responsible and causes a disease, and until we have a copy of it cloned, we can't think about gene therapy.
One of the major benefits that will come from the Human Genome Project is it will give us a detailed map of the 23 pairs of chromosomes. And these, in turn, will allow us to identify, to chart the genes that are involved in many different diseases that range from Alzheimer's disease to manic depressive disease to various types of cancer.
Deciphering the human genome may well become one of the most important discoveries of our time, as powerful as splitting the atom. But with this power comes a risk. Scientists are now on the brink of manipulating DNA, the blueprint of life itself. Are we as a society ready to manage such awesome power wisely?
The public has genuine concerns about the intentions and implications of scientific discoveries. They have particular concerns because in the past, some of their fears have been justified. The public was told that harnessing the atomic energy and nuclear energy was going to be extremely beneficial. But at the same time, it was harnessed into making an atomic bomb and a hydrogen bomb. So I believe the public's fears are not groundless.
What if we have a cloned piece of DNA that does something we don't even understand completely? Somehow it gets out in the environment and it does something completely unexpected or unknown. it's always a possibility. When you can change the genetic makeup of any organism or any species fairly easily and readily, I think it's hard to predict down the line exactly what the consequences will be. I think it's a responsibility of scientists doing the work to try and understand and think about these kind of problems, but I think it's also very healthy and good that there are organizations and people who keep the pressure on to think about these things and make us be concerned and skeptical, even if we think we don't have time to be.
You can always come up with the scenario of the mad scientist creating monsters or whatever, but I don't think that should stop us from trying to help kids with bad diseases. And I think that's our focus. Our focus is there are some bad genetic diseases, and if we can help provide new treatments for those, that we would be morally wrong not to do that.